Alright, so in this video we're going to get started with our next unit, so unit 2, which is cells. And we're going to start out today just with some basic stuff because I know that you are taking an assessment today from Chemistry of Life, so I don't want to give you too much other things to do. So in this video we're just going to talk about um, the different types of cells, so prokaryotic and eukaryotic cells. um, the history of where they came from, and their similarities and differences, and then answer the question, why are cells so small? So, for example, why can't cells be as big as a baseball, or the size of a golf ball, or something like that? Why do they have to be the size that they are, which are so microscopic, right? So let's get started. So basic cell theory says that all organisms or living things are made up of cells. And it's the basic unit of a living organism. Okay, it's the smallest building block that you can't divide any farther. Okay, and still get something, you know, that would have the same properties as a cell. Okay, and then the last part of the cell theory says that all cells come from pre-existing cells. So a given cell can reproduce and make new cells. So these are the three basic ideas that govern cell theory, or what we believe to be the properties of cells. So cells are important for everything that an organism needs to do. So for example, reproduction, growth and development, using energy, responding to uh, external stimuli from the environment, maintaining homeostasis or the narrow balance of conditions necessary for life. All of these things um, have to be done at the cellular level. So cells are responsible for making sure that the organism that they're part of as a whole can do these things. All right, and we'll talk about as the unit goes on how cells accomplish each one of these things to help the organism that they're part of survive. So some characteristics of all cells. So it doesn't matter what type of cell they are, all cells have these same characteristics. All cells are surrounded by a plasma membrane. All right, and we'll talk more about the plasma membrane, specifically its structure and function in a different video. All of them are filled with cytosol, which is kind of like a fluidy jelly type of substance that's inside, all right, that the different part things of the cell float around in. All cells contain genetic information of some sort, like contained in chromosomes, and the way those chromosomes might be structured might be different depending on the type of cell. And all cells have ribosomes, which are tiny organelles that make proteins from the information contained in DNA. So these things are true of every type of cell, whether it's a eukaryotic cell or a prokaryotic cell. Now, going off of that, there are two types of cells. So all living things that are alive are made up of either prokaryotic cells or eukaryotic cells. All right, prokaryotic cells, um, the DNA is stored in what we call a nucleoid region, and it doesn't have a nucleus. So prokaryotic cells do not have a nucleus, and their DNA is just in the cytosol in this structure called um, a nucleoid. Whereas eukaryotic cells contain DNA in a chromosome or chromosomes that are inside a membrane-bound nucleus. There's a nuclear membrane, and inside there is the DNA. And that's actually where we get these terms. Eukaryote or eukaryotic means true nucleus. True nucleus. So they have a nucleus, and prokaryotic cells do not. And that's one of the big differences between these two different types of cells. So for example, here's a prokaryotic cell. look at it and just see kind of what you notice about it versus a eukaryotic cell. This one looks a lot simpler. This is a lot more complex. And this one you can see there's a whole bunch of different stuff inside of it, where this doesn't seem like it has nearly as much interesting stuff inside of it, right? Um, Part of the reason why is prokaryotic cells are about 1 billion uh, years older than eukaryotic cells. So prokaryotic cells are definitely older than eukaryotic cells, and we'll talk about how we got eukaryotic cells in a little bit. So here is an example of a prokaryotic cell. It's definitely much simpler in structure than a eukaryotic cell, and it lacks a nucleus and other organelles that are enclosed in membranes. So here you can kind of see the basic structure of a simple prokaryotic cell. And then eukaryotic cells, on the other hand, are much more complex. Within the cytoplasm, they have a bunch of different organelles that are specialized structures that each do a specific job. All right, they each do something specific to help the cell maintain homeostasis. Those same functions that the organelles in a eukaryotic cell do still need to be carried out in a prokaryotic cell, but they aren't localized to a specific region within that prokaryotic cell. Those chemical reactions just are carried out everywhere in the cytoplasm. And because of this, eukaryotic cells can be much bigger than prokaryotic cells. Much bigger. So here is an example of a eukaryotic cell, and you can see all of the different membrane-bound organelles that are inside of it, that do all of those specialized functions that we talked about. And here's another one. This is a plant cell. So let's kind of look at the differences. Here's a animal cell, and you'll notice that it doesn't have things like chloroplasts or a cell wall or a central vacuole. These are things that we'll talk about when we talk about the structures of eukaryotic cells. And here's a plant cell that you see don't have lysosomes or centrioles or flagella that animal cells do, and we'll learn more about what each of these organelles do in our next video. So if we're going to make it into a little chart about prokaryotes versus eukaryotes, this is what I would do. And so this is something that you should have a good idea, not exhaustively being, I don't expect everybody to draw this on a piece of paper and be able to, fill this in from memory from blank, but you should have a good idea after today about the differences between eukaryotes and prokaryotes in some or most of these. So maybe just pause here for a minute and kind of look over this, see which ones you have remembered from a previous and which ones you'll need to try to commit to memory. So going from there, I said one of the things we want to talk about is how eukaryotes ever came to be, because we said prokaryotes are about 1 billion years older than eukaryotic cells. So the generally accepted theory about the origin of eukaryotes is called the endosymbiotic theory. And this theory roughly says that a long time ago, prokaryotic cells engulfed other prokaryotic cells by endocytosis. And this word on here, endocytosis, we're going to talk about that a little bit later in this unit. And this resulted in the first eukaryotic cells. So eukaryotic cells have different organelles because they are remnants of previously engulfed prokaryotic cells. This theory was first proposed by Lynn Margulis and explains the origin of eukaryotic cells and of certain membrane-bound organelles that we find in eukaryotic cells. So if we were going to look at what actually happened, we might look at this picture. So a long time ago, there were just prokaryotic cells, and some prokaryotic cells were able to develop special functions through evolution. Some were able to, for example, develop the ability to break down uh, external chemical substances and to create chemical energy. Okay, they were able to go through cellular respiration, and they were, able to produce a lot of energy on their own. And so other ancient prokaryotic cells kind of engulfed them like this, so that they could also harness that energy. And over time, those ancient heterotrophic prokaryotes merged with and became fused with primitive eukaryotic cells. Okay, and the same thing happened. Okay, modern chloroplasts arose from ancient prokaryotic cells that had evolved photosynthetic abilities and had likewise become engulfed by these primitive heterotrophic eukaryotes and turned into all plants and plant-like protists. So chloroplasts and mitochondria are two organelles that used to be, in ancestral times, separate prokaryotic cells. So mitochondria are membrane-bound organelles that produce energy for the cell, and chloroplasts are membrane-bound organelles that capture light energy for plant cells they use to make food. All right, and they used to be separate prokaryotic cells that eventually were engulfed by and incorporated in fledgling eukaryotic cells. Now, there's a bunch of evidence to support this idea, and some of it is that there are great similarities between mitochondria, chloroplasts, and other prokaryotes, such as each containing their own circular DNA, having their own ribosomes. So, chloroplasts and mitochondria have their own ribosomes. They can divide by binary fission, just like prokaryotes. So these three things are commonalities between only mitochondria, chloroplasts, and prokaryotes. No other organelles have these things. And so it's a support of this theory and this idea. The last thing that I want to talk about is why cells are the size they are. Why are they so small and not bigger? So there's a lower limit kind of to the size cells can be. In the lower limit, the smallest bacteria that are seen, mycoplasmas, are about 1 micron big, right? Most bacteria are between 1 and 10 microns. If they were any smaller than that, there just wouldn't be enough space to accomplish all of the things necessary for life. But there's also an upper limit, all right? The eukaryotic cells, which are bigger than prokaryotes, are between 10 and 100 microns, so about 10 times bigger than the biggest prokaryote, right? And there's not really any bigger than 10 microns, all right? And the idea is, why can't there be any larger than that? And the idea is that there's something that limits cell size, and that is what is called the surface area to volume ratio. And that's the idea that as cells get bigger, the volume inside of the cell increases faster than the surface area of the cell. So a smaller object has a greater surface area to volume ratio. Okay? Um, the idea, okay, and you can see right here as the surface area to volume ratio increases, as the surface area to volume ratio changes between a cell as it gets bigger. So as a cell gets bigger, its surface area to volume ratio decreases. Now, why is surface area to volume ratio important? Well, as volume increases, there's more stuff inside a cell that needs nutrients, like oxygen and glucose and things like that. But as a cell gets bigger, there's less of a plasma membrane around its surface to allow substances in. So as a cell gets bigger, you have a much greater increase in the amount of material inside of that cell that needs nutrients to come in and needs waste to get out, and a lot less space over which those nutrients and that waste can pass. So after cells get a certain size, any bigger wouldn't be able to be sustainable. We couldn't get nutrients in fast enough and waste out fast enough to maintain life. All right, and so the surface area to volume ratio is really important. The bigger it is, the more efficiently waste and nutrients can be transported in and out of the cell. And the lower it is, the harder it is for a cell that size or larger to stay alive. Right? And we calculate surface area to volume ratio just by taking the surface area of that cell and dividing it by its volume. So like I said, the metabolic requirements, needing nutrients in and to put waste out, sets an upper limit for cell size. And large cells, they can't move materials in and out of the cell fast enough to support life. All right, we need more nutrients inside of the cell because there's more volume to maintain as healthy, but we don't have the same ratio of plasma membrane to allow things to cross as in a smaller cell. So it becomes harder and harder to support life the bigger and bigger cells get. So, how do we get bigger organisms like us? And the answer is to become multicellular, to divide, because now each of these cells has a high surface area to volume ratio and can exchange nutrients and waste with the outside environment extremely fast and maintain life. So that's going to be it for this video. Please let me know if you have any questions. Otherwise, I'll see you in the asynchronous session tomorrow.